everyone. I am Professor Charu from Ramjas College, University of Delhi, and I am continuing with our series on Microbial World, in which I am talking about the overview of microbiology and introductory aspect, where we are discussing about the history of microbiology and the key contributions of renowned scientists. We discussed about the contribution of Edward Jenner, where the first vaccination and the vaccines during that time was developed, and then we also talked about the antibiotics which was discovered, the penicillin which was discovered by the Alexander Fleming. So, the discovery of Alexander Fleming of this antibiotic opened the second golden era of microbiology. So, after that, there was series of things that were developed, and now basically the association and discovery of DNA has also been started. So, there were many, different antibiotics for many different organisms, started being discovered. As I said, the vaccination, the vaccines against various different diseased organisms was also considered. So, the contribution again, all of these scientists are Nobel laureates. So, again it is given on your screen that this formed the second golden era of microbiology which was there. So, what happened was then came the third golden era, when they basically said that everything is associated, and obviously the techniques and tools such as genomics and gene sequencing, like Sanger came in, and there was the genes could be sequenced, and all those things also revolutionized the world of microbiology, because they emphasized on the fact that there are these invisible creatures which is basically ruling a lot many things. So, not just the diseases, they started finding diseases. As I said in my first session on Microbial World, the diseased organisms or pathogen, they are a very small amount of the entire microbes which is there on this planet. Many more are the advantageous or beneficial uses of these microorganisms what they are providing to us. So, pathogens, but obviously they caught our eye or they start these discoveries because of the treating various kinds of diseases. So, the contribution of these scientists are again very important. On the screen there is this Professor Tu, which is there, who discovered artemisinin, which is working from a Chinese sage plant, and that compound he isolated, which works against the malaria as well, the malarial parasite as well. So, lot many discoveries, and that is called as the third golden era of microbiology. Now, before going to discuss about the microbial diversity, so these are some of the important contributions that we talked about. Now, we move on to introduce the various kind of microbial diversity. Microbes are enormous, that I think by now you appreciate that. We talked about that, they are ubiquitous, they are pervasive, they are present everywhere, and I also discussed that, less than 1 gram of a virus put the entire globe on its knees. There was only less than 1 gram of infection. So, you can imagine the mighty power of these microbes, even less than 1 gram. What is 1 gram, right? So, that less than 1 gram amount of virus made the entire earth stand still, the coronavirus I am talking about. So, before I go to talk about the different variety or the diversity of these microbes, it is very important to classify them, to put some kind of an order into it, to understand them better. Generally, what they did in earlier times was they categorized them or classified them based on the morphological features, because what they could observe or their habitats, or where they dwell and what kind of behavior they had. So, based on that they classified or categorized these organisms. So, I hope you understand why this biological classification is very necessary, because if you start studying individual organisms and taking out, delineating all the characteristics of individual organism, it is very tedious and it is very exhaustive. So, if you categorize the organisms based on their relatedness, then it becomes easier, because one representative if you have studied, you may think that the others also behave like that. So, but obviously initially it was not like that, because they were only based on the morphological characteristics or only based on the habitats. So, some of the properties could be delineated, but not many. Nowadays, it is basically the genetic relatedness, which also relates to the evolution, which is the phylogenetic, that how they are coming from. So, now we talk about the relatedness at the DNA level, at the nucleotide sequence level, and then we try to classify them into different categories so that they can be learnt easier. It becomes easier to learn and as well as easier to communicate with each other, so that we can talk and communicate the research among globally, and therefore comes and finds more and more newer things or reach conclusions. So, this taxonomy is very, very important. So, before I talk about the microbial diversity, let us go through the taxonomy, that how, again the history of taxonomy, that what were the different kinds or methods of classification that came into being as newer and newer things, again related to the history of microbiology as well. So, I thought it would be better to understand this first before diving into the microbial diversity, which is very vast. So, the dawn of order basically started when the organisms are put into various categories, or what is called as the taxa. The singular word for that is a taxon, to show the degrees of similarities among them. So, you understand the similarities, which is basically the relatedness. So, the degree of relatedness, based on that we put them into different taxa. These similarities are due to the relatedness, that all organisms are related through evolution. So, it is coming from a single organism, so all organisms are basically related through evolution. So, that is an important aspect. Two other terminology that you will come across, and before I move further, I just want to clarify that one is the systematics and another is termed as the phylogeny, or basically the same thing, which is the study of the evolutionary history of organisms, and the hierarchy of taxa reflects their evolutionary or what is termed as the phylogenetic relationships. So, it is very important. Now, we just do not talk in terms of the related characters that we are seeing now, but we discuss the relatedness over the centuries, that where they are coming from. So, more similarities in their DNA in the past, from the past organisms or the evidences that we have, such as fossil evidences and other evidences, then we can talk about that probably they are coming from the same ancestor. And such kind of an hierarchy reflects their evolutionary or phylogenetic path. So, we study it under systematics. So, taxonomy, systematics, phylogeny, these are the words that you will come across a lot, and so I hope that is clear to you what they are. Now, this classification started in circa 384 to 322 BC, when the Aristotle's time, he was the philosopher of nature. He was the first great biologist who applied the systematic observation to the natural world. Obviously, there are so many organisms, but he was the first one who stated that let's talk them about in two different things. There are two distinct categories that we can observe, that there are certain which are termed as animals and there are certain which are termed as plants, usually isolated by the sedentary thing, that the plants are sedentary and the animals are moving. That is the basic characteristics. Now, there are otherwise also, but that was the initial thing, that these are animals, these are plants. So, he was the first one who started studying the organisms or put through these things, that they need to be categorized as animals and plants. He authored what is called as the Historia Plantarum and Historia Animalium, cataloging over 500 plant species and numerous animals with remarkable detail. He classified the organisms based on observable characteristics, growth form such as tree, shrub, herb, and animal traits such as viviparous or oviparous reproduction. So, certain characteristics which were observable, whether they were tree or whether they were shrub or whether, and there were certain characteristics, they have woody trunk, they are tall, they are short, they are creepers, they are whatever. So, they identified the observable characters and certain like viviparous and oviparous reproduction, that whether they lay eggs or whether they are giving birth to human beings. So, he classified the organisms based on these observable characteristics, and he was the first one to start with this two kingdom, as we say, classification and categorize the organisms into plants and animals. Then came Carl Linnaeus. He is the father of taxonomy, and he proposed two very important things. One is, we know him, that he has given that binomial nomenclature of the scientific, what we call as the scientific names. And the second thing is he also proposed the hierarchical classification, that there is a broader, all the organisms are together, and then based on the relatedness we keep clubbing them into different categories. So he gave that hierarchical biological classification, and he is termed as the father of taxonomy. He introduced a formal system of classification with two kingdoms, which is the Plantae and the Animalia. He developed the binomial nomenclature, as I said, that the genus name and the species name, and then there are certain guidelines that or the rules that need to be followed, but he allowed the scientists worldwide to communicate precisely about organisms. As I said, in the research or in the scientific community, communication is a very important aspect. If you discover something and you just keep it to yourself, it is of no use, because everybody does not need to invent the wheel again and again. But if you discover something, a process, a mechanism, an organism, and you put it across onto the globe, then everybody knows and the people start working forward from that. So again, this is very, very communication. Scientific communication is very important in the scientific community or the research process. So he developed this binomial nomenclature, which was approved universal. Every organism will have this kind of names, where the first name will represent a genus and the second name will represent the species. So then came Ernest Haeckel, and he proposed the kingdom Protista, because what happens was that, again, observation, that they observed, as we had discussed in the history of microbiology. Now the microbes were starting, the scientists started discovering microbes. They associated the diseases with the microbes. So the invisible creatures were also very important. So they could not be put them with the Animalia or Plantae, because they were all invisible creatures. So Ernest Haeckel proposed this third kingdom, which is called as the Protista, to include all these bacteria, protozoa, algae and fungi, which were basically unicellular, or which were basically not visible to the naked eye. And therefore came the three kingdom classification. Obviously we know that this three system was not enough even, and we cannot club the bacteria and fungi together. But at that point, the relatedness point was basically that they were not visible to the naked eye. And as more and more organisms were discovered and their association with the harmful effects or useful effects were discovered, he proposed a separate kingdom, which was the Protista. Then came Whittaker. He founded what is called as the five kingdom system. He said that the prokaryotes were placed in the kingdom Prokaryote or Monera. So an important observation came in here. So what they did was, what they observed was that there were two different types of even single celled organisms which were there. The one which were very simple in their appearance and organization. They did not have a lot of compartmentalization which was present there. And certain had all these kind of compartmentalization which was there. So membrane bound structures which were present inside the cell. So obviously you cannot categorize them together. So prokaryote they termed as the primitive ones, that they contained a primitive kind of a karyon is basically the nucleus, because that is a very prominent organelle inside a cell which is visible when you see it under a microscope. So they observed that some were not having it and some was having it. So he basically founded this five kingdom system, that no, you cannot club both of them together. So we need to separate them out. So he called that whatever organisms did not have this well structured organelle inside the cell, he termed them as kingdom Prokaryota and classified them under the category Prokaryota or what is Monera. And then all other organisms which contained a well defined structure called as the nucleus later, he comprised the other four kingdoms. And what were the other four kingdoms? Protista, Animalia and Plantae, which were already there, and he separated the fungi, because fungi were again entirely different. He could not place them under plant, although many years, many centuries they were considered as plants only. But then they observed that they do not fit in with the plantae, and therefore they separated into a separate group. Of course, some of the characteristics, they are matching, but still they have that potential to be called as a different kingdom. So, H. Whittaker founded this 5-kingdom system classification in which all the prokaryotes, the primitive or the simple, the bacteria and everything which came in to come under prokaryotes or monera, and the eukaryotes contain the 4 kingdoms in which there were protista. So, protista became basically the eukaryotic microbes which were there. So, all those were put under protista, and then there will be amoebae etc., unicellular ones which were there, and then animalia, the plantae and the fungi. So, that is how the 5-kingdom system classification came into being. Then came the Carl R. Weiss. Now, Carl R. Weiss proposed a level above the kingdom. So, now we are talking about the the starting with a very basic Aristotle just 2 category thing, which is the animal and plantae, and then came the kingdoms. So, we call the animal kingdom, the plant kingdom and the protista, which became separate. Then came the 5-kingdom classification. So, these kingdoms we were relating based on the relatedness which was there, and subsequently over time when you observed, when the scientists observed that there were differences in the organisms which were contained in a particular kingdom, that they can form a separate kingdom only. Now, what did Carl R. Weiss suggested was that what they observed was that the prokaryotes were very primitive. So, they now talked about how the cells originated. By that time, the Darwin was there and all those people came in, and if you see the history, then what happened was they observed that there was this kind of a nucleoplasmic cell which was there, or a plasmic cell which was there. And initially there was no compartmentalization, and then what happened was that gradually some cells started having this kind of a compartmentalization for efficiency of functioning, of course. And as the organisms become unicellular, say multicellular, multicellular from unicellular. So, that way these kind of and eukaryotes came into being. And then obviously there were certain exceptions which were in between, and then gradually when they moved on, then what they observed was that on the basis of certain structures, when the tools and techniques were developing, when they identify at the level of genetic components, they observed that the cells were not just prokaryote and eukaryote, but there were certain cells which were neither prokaryote nor eukaryote. And the one gene that they observed was the ribosomal RNA gene. Now, you know what ribosomes are. They are the protein synthesis factories which are present, and we have talked about in our cell biology series that proteins are formed on these ribosomes. In these ribosomes there are 2 subunits which are present, and there are genes specifically encoding the rRNA which get associated with protein molecules to give rise to these subunits, and all those kind of things. And these subunits are recognized by differences in their sedimentation rate. So, when you centrifuge it, then the rate at which they sediment, they are recognized as different subunits. So, when they observed the different kind of cells, the cells were not just 2 types, which is the prokaryote or the eukaryote, but the prokaryote, eukaryote and another type of cells. There were 3 types of cells which were having different types of rRNAs associated with them. And therefore, Carl R. Weiss basically took these 3 types of cells a level above the kingdom and called them as domain, which is based on the nucleotide sequences of rRNA. So, I hope I am making clear myself that how did this domain came into being. So, initially there were 5 system classification, initially there were 2 categories, then came 3 kingdom classification, then came 5 kingdom classification. But then what he observed was that for the 5 kingdom classification, the major distinction was made between the prokaryote and the eukaryote. So, there was a 1 kingdom which was prokaryotic kingdom, and the rest 4 kingdoms were the eukaryotic kingdom. So, the type of cells, there are 2, the prokaryote and the eukaryote. and they differ of course because of the presence of these cell organelles etc., but also differ at certain different things, the kind of cell wall or cell layering they had, and then as I said they differ because of the presence of the different types of ribosomes which were there. And these ribosomes contain rRNA, ribosomal RNA, which is encoded by certain nucleotide sequences or genes in the DNA which is present. So, when he was observing, then they observed that there are not just 2 types of cells, but there are 3 types of cells. And in these 3 types of cells, these rRNA differ. So, how can we just have 5 system kingdom classification based on the division of prokaryote and eukaryote? And therefore, he proposed a level above the kingdom, which is basically termed as the domain. So, I hope I made myself clear on this domain aspect. So, he gave what is called as a 3-domain system, talking to the level of 3 cells as we say. Animal, plant and fungi are the kingdoms in the domain eukarya, and different prokaryotes under the domains domain bacteria and the domain archaea. So, the eukarya contained all the animals, plants and fungi. Now, what we were calling as the prokaryotes, now that prokaryotes are all not same. There are certain prokaryotes which contain different rRNA genes, and there were many other characteristics like they were found at different special extreme habitats, or they had different peptidoglycan layer etc. So, all these distinction in the features prompted them to create the 3rd domain which is called as the domain archaea. The domain bacteria includes all of the pathogenic prokaryotes as well as many of the non-pathogenic prokaryotes which are found in soil and water. Even the photoautotrophic prokaryotes are also in this domain. So, there was a domain which is eukarya containing the animals, the plants and the fungi, and then in the prokaryote there was a domain bacteria that includes all the pathogenic prokaryotes as well as non-pathogenic prokaryotes, which are generally found in soil and water, normal soils and water, everywhere around us as we say, and even the photoautotrophic prokaryotes. Now, this domain archaea, this includes the prokaryotes that do not have peptidoglycan in their cell walls. They often live in extreme environments and carry out unusual metabolic processes. So, these features did not correlate at all with the domain bacteria organisms. So, they created a separate domain because of the cell type as well, because they contained a different kind of peptidoglycan. They do not have peptidoglycan in their cell walls. They often live in extreme environments. What are the extreme environments? They were methanogenic bacteria. Methanogenic bacteria are obligate anaerobes. They do not require oxygen for their growth, and they produce methane by using water and carbon dioxide. Then there was there are what are called as halophiles, which are basically they live in highly salted environments, or there are extremothermophiles, which basically can survive the boiling water. You must have observed lot of hot water springs. There are in India, there are many hot water springs. One is at Manikaran, then there is a Kheer Ganga hot water springs. Near Delhi there is Sona, where there are hot water springs. So, these hot water springs are natural bodies from the earth the hot boiling water is coming. And for your astonishment, this hot water boiling springs also contain lot of bacteria which is present there. And obviously, because they can withstand these high temperatures, they have special kind of machinery. They carry out unusual metabolic processes which is there. Their cell walls are entirely different, and therefore they are categorized under the domain archaea which is there. So, this is the 3-domain system what Carl Weiss basically proposed, and it has been in use till now. That initially there was a general cytoplasmic cell which was there, a nucleoplasmic cell, and gradually these started growing. Now, you can also observe that this nucleoplasmic cell, there was a horizontal gene transfer that was occurring. So it was not like that, that there was not a distinction between these horizontal, very clear distinction between these domains. There used to be exchange of genes that occur. But then there was a bacterial domain where you contain all different kinds of bacteria which is there, and some of these bacteria became endosymbiotic to early growing eukarya, and therefore the typical eukaryotic structure appeared, where we have mitochondria and chloroplast, and that mitochondria and chloroplast in eukaryotic cells there is an endosymbiotic theory, that once they were free living bacteria, they were engulfed by the primitive eukaryotic cells, and therefore they became to function in a particular aspect and gave rise to the current eukaryotic cells. And in the archaea, you can see that there are thermophiles, there are halophiles, and there are methanogenic bacteria which is there. So this is the 3 domain system which we basically use. The second aspect which is very important is the taxonomic hierarchy. All organisms can be grouped into a series of subdivisions. So we categorize them, but how do we ascertain that in that group classification we basically create a taxonomic hierarchy which is there. For example, we start from an individual, in individual will form the related individuals, the closely related organisms that breed among themselves, they are termed as eukaryotic species. So we call them as species because they can interbreed amongst themselves. Then now these species, they differ from each other in certain ways, but are related by descent. Descent means evolutionary history. So they come under together what is called as a genus. The related genera make up a family. A group of similar families constitutes an order, and a group of similar orders make up a class. The related classes in turns make up a phylum. All the phyla that are related to each other make up a kingdom, Animalia, Plantae, Fungi, Bacteria, and Monera and Protista, the 5 kingdom classification. But then further up, they are related kingdoms are grouped into a domain, that is the bacteria or the archaea or the eukarya. So this is basically how the classification occurs, that we have all different organisms on the top together, and then we keep doing the related organisms if we go from the top, or basically if we go from down to up, you can see there is E. coli which is there, and this E. coli related species which can breed amongst themselves will come under E. coli. Now E. coli basically this bacteria which cannot breed among themselves will form the genera, and then further up and they will further constitute the entire organism which is there. So I hope you understand this kind of hierarchical classification as well as a little bit about the history of classification, because in our next session then now we will talk about that under these different categories how diverse the microbial environment or microbes are, so that you can appreciate them more. There are so many what kingdoms are there, what classes are there, what families are there, what genera are there, what species are there. So there are so many microorganisms which are there, and we will talk about this microdiversity by learning them under different categories or having an ordered study which is under the biological classification. So we continue in our next sessions. Thank you for your attention.